If we're touring the cell cytoplasm, one of the more interesting structures is the endoplasmic reticulum, or ER. The ER is a net-like organelle that's continuous with the nuclear membrane. Like the nuclear envelope, the ER is enclosed by a double membrane, and this membrane invaginates to form cisternae that greatly increase its surface area. There are two flavors of endoplasmic reticulum, rough and smooth. The rough ER is something like a 5 o'clock shadow, created by ribosomes peppering its surface. Well, what are ribosomes? Glad you asked. Ribosomes are protein RNA complexes that are responsible for synthesizing proteins. And a human cell might have as many as 10 million ribosomes working hard to make all the proteins your cell needs. Ribosomes bound to the rough endoplasmic reticulum feed their newly synthesized proteins directly into the rough ER, where the proteins are folded into their 3D shape with the help of chaperone proteins. In the endoplasmic reticulum, proteins can be modified by the addition of carbohydrate groups, which is called glycosylation, proteolytic cleavage, the creation of disulfide bridges, or the addition of other functional groups. You'll learn more about some of these modifications elsewhere, but for now, it's just important to know that they exist. Proteins synthesized on membrane-bound ribosomes are typically destined either for secretion outside the cell or to be embedded in a cellular membrane. In contrast, free ribosomes, floating freely in the cytoplasm, synthesize proteins that have an intracellular destination, such as cytoplasmic proteins involved in cellular metabolism. The smooth-shaven cousin of the rough ER is the smooth ER, which is not speckled with ribosomes. The smooth ER plays important roles in lipid metabolism, steroid hormone synthesis, and detoxification. And since the liver is the central site of detox in the body, liver cells tend to have an extensive smooth ER. As part of lipid metabolism, the smooth ER produces the phospholipid components of cellular membranes. The smooth ER in muscle cells is specialized to store calcium for muscle contraction. So, in muscles, the smooth ER is referred to as the sarcoplasmic reticulum. Once proteins are synthesized, they're packaged into vesicles, destined for the Golgi apparatus. You've probably heard the Golgi apparatus referred to as the post office of the cell once or twice or 5 billion times. And it's a little cringeworthy, but that's approximately the goal of the Golgi apparatus. The Golgi is composed of stacks of pancake-like chambers known as cisternae, similar to those in the ER. It receives protein-packed vesicles at its cis face, modifies them internally, and then packages them into membrane-bound vesicles that bud off from the transface to be sent to their ultimate destination, typically toward the plasma membrane for secretion outside the cell, or some other cellular membrane. Next, lysosomes can kind of be thought of as the garbage disposal system of the cell. Another cringeworthy analogy, but it makes it easier to remember. Basically, extracellular stuff that needs to be broken down enters the cell through endocytosis, where a vesicle pinches off from the cell membrane, and then that vesicle fuses with a lysosome inside the cell. In addition, intracellular debris in the cytoplasm can become enclosed within a vesicle that then fuses with the lysosome for degradation. And this processing of cytoplasmic waste is called autophagy. Lysosomes are membrane-bound vesicles that pinch off from the Golgi apparatus, and they house hydrolytic enzymes that chop up and digest everything from macromolecules to retired organelles to foreign substances. Now, what if one of these lysosomes went rogue and released its digestive enzymes into the cellular cytoplasm? Code red, right? Well, evolution thought of that. Lysosomal enzymes function best at an acidic pH, and therefore the lysosome interior is kept at a pH of about 4.5 to 5 or so. So in the event that a lysosome were punctured and spilled its contents into the much more neutral cytoplasm, well, those enzymes aren't going to be able to do much. The last organelle of importance is the peroxisome. As its name implies, the peroxisome accumulates and neutralizes peroxides, like hydrogen peroxide. If you've ever doused a wound with hydrogen peroxide, you know that it's a pretty powerful chemical. And in fact, hydrogen peroxide generates reactive oxygen species that are harmful to the cell. In addition to protecting against oxidative stress, peroxisomes have a critical role in breaking down very long-chain fatty acids via beta oxidation. As you can tell, eukaryotic cells are highly compartmentalized, permitting division of labor by membrane-bound organelles. Furthermore, different cell types may have their own specialized organelles to facilitate a particular function. But the overall template, nucleus, mitochondria, membrane-bound organelles, that remains the same.